Hi everyone, I hope you all have a good Labor Day holiday. We'll talk about Mendelian inheritance today. I know that you probably start to learn this topic since middle school and already very familiar with it. But as Mendelian inheritance is the most fundamental knowledge of human genetics, I would like to go over this again. But there is a higher requirement for you. I'll focus on problem solving and we'll combine this with meiosis process so we can remember this much better. In our textbook, trait means any observable property of an organism. In the past, people had always known that traits are passed down. They knew that the young living things like, but not exactly like their parents. But no one really understood how it worked until Greg Mendel, an Austrian monk, began to experiment with pea plants in his garden. From 1856 to 1864, Mendel grew thousands of pea plants, observing the parent plants, their offspring, and generations that followed. A life cycle of garden pea is about 3 to 4 months. So Mendel could take 3 generations in a year, which is a lot faster than using mice. He was using mice when he began his research at the very beginning. But then if he used the mice, he probably would not be able to make such a big discoveries because most traits in mice do not controlled by single genes. Mendel studied the inheritance of 7 different features in peas, including height, seed color, and seed shape, and flower positions. To do so, he first established the pea lines with 2 different forms of feature, such as tall versus short height, or round versus wrinkled seeds. This diagram shows one of Mendel's crosses. True bleeding strains of pea plants with smooth and wrinkled peas were used as the P1 generation. Interesting, all the offspring in the F1 generation had smooth peas. Self-fertilization of F1 plants gave rise to plants with smooth and wrinkled peas in the F2 generation. About 3/4 of the peas were shown as smooth, and about 1/4 were wrinkled. So the ratio is about 3 to 1. Interesting, on all 7 traits that Mendel studied, the crosses give Mendel the same results. On average, 3/4 of the F2 offspring showed 1 trait, and 1/4 showed the other trait. So always keep this 3 to 1 ratio. Mendel called the crosses that involve a single trait as monohybrid crosses. One form of a feature, such as tall, always concealed the other form, such as short, in the first generation after the cross. Mendel called the visible or this expressed trait as a dominant trait, and the hidden or the unexpressed trait as a recessive trait. He found that although pea plants have 2 factors for each trait, they contribute only 1 to their offspring. Moreover, each plant carries 2 genetic factors for each trait, 1 from each parent. So what are the genetic factors he mentioned about? Are they DNA, chromosome, or genes? You can think about it. So also, traits were not blended as they passed through parents. Okay, now here's an explanation on what Mendel had observed, okay? With the further development of modern genetics, scientists define observable properties of an organism as a phenotype, okay? Trait, for example, is a phenotype. So specific phenotypes in human can include blue eyes, long fingers, curly hair, dark skin, etc. In contrast, the specific genetic constitution of an organism is named as a genotype. For example, we learned that RB deletion mutant like serine 61 mutated RAS gene, etc. Showing on this picture are the phenotypes and genotypes in peas. Like for instance, the smooth and the wrinkled seeds are phenotypes, but the genotypes has 3 different combinations. For example, the genotypes can be 2 big S, 2 small s, and 1 big S and 1 small s, indicating that phenotype and genotype are not always correlated. Gene alleles are the genetic factors Mundell mentioned in his conclusions. Allele is one possible alternative form of a gene, usually distinguished by its phenotypic effects. So what does this really mean? From our first class, we learned that the gene is a piece of DNA that can encode a protein, right? So allele is a gene localized in homologous pair of chromosomes, all right? So their DNA sequence can be the same or different. That is the reason why one allele can make up, like, say, purple flowers, and the other allele can make white flowers. It is the encoded protein that determines the color. Where does the homologous pair of chromosomes come from? Yes, one can come from your dad, as a paternal, and one from mother, we call it a maternal, okay? You need to understand all this concept before understanding Mundell's law at a molecular level. And please don't confuse allele with the sister chromatids, okay? Sister chromatids are replicated copies. Therefore, the gene sequences or DNA sequences are exactly the same. But the allele can be same or different. Now let's take a look at what homozygous and heterozygous mean. This is actually easy to remember. Homozygous means that 2 alleles are identical. Okay, so here the identical means the sequence is identical. Okay, so heterozygous means the 2 alleles are different. Again, this different means the DNA sequences are different. And that is the basis why the encoded proteins are different. In this picture with these 2 different alleles, there could be 3 possible combinations with 2 homozygous. 1, both are big A, and 2, both are small a. And there also can have 2 heterozygous, but if the heterozygous, they are actually the same, 1 big A and 1 small a, all right? So let's go over Mundell's law together and see how these laws can be explained by meiosis we learned from last lecture. Mundell's first law is the principle of segregation, meaning individuals carry 2 alleles for each trait, but only pass 1 allele onto their offspring. But how is this possible? Let's think about the separation of 2 alleles. When does it happen in meiosis? Is this in meiosis 1 or meiosis 2? If your answer is meiosis 1, congratulations. This means you remember and understand how this meiosis works. A Punnett square diagram can illustrate this principle as our next slide shows. So this is an example showing how Punnett square can be used to derive the F2 ratios in a cross from the F1 generation, okay? Say the F1s are heterozygous, and the bigger S is a dominant trait, means smooth, and the small s means wrinkle, which is a recessive trait, okay? When they cross, based on the principle of segregation, all right? So each parent will contribute a big S allele and a small s allele, okay? And these alleles have different combinations that can be deduced from the Punnett square, resulting 2 different homozygous, big S or small s, and the 2 identical heterozygous offspring, both big S and small s, okay? Because smooth is the dominant allele, So phenotypically, the two heterozygous offsprings also show smooth phenotype. In exams, they always have questions asking about the ratio between different phenotype or genotype, okay? So you have to pay attention to what the questions ask so that you can give the right answer. Mendel's second law is the principle of independent assortment. Members of one gene pair, meaning the alleles, they're separate or assort independently of other gene pairs. So the context of Mendel's second law is that you need to have at least two traits, and these traits won't affect each other. So when would this happen in meiosis? Mendel's observations about the segregation and independent assortment can be explained by the behavior of chromosomes during meiosis. Okay, the random arrangement of maternal and the paternal chromosome pairs at metaphase of meiosis 1, see here, produces a 4 combination of the 2 gene in gametes. 4 combination, 1, 2, 3, 4, okay? So both laws are determined in meiosis 1. Punnett square can also be used to calculate the two traits. For instance, this picture shows the dihybrid cross. So can you tell the two pairs of traits in this picture? Yes, one pair is the smooth and the wrinkle, right? So, and the other pair is yellow or green. These two pairs of traits are sorted independently. Therefore, even though the P1 plants are different, they can generate the same F1 plants, right? So how can you determine the dominant traits here? Yes, F1 plant has the dominant traits. Here, smooth P is the dominant traits over wrinkled P's, and yellow P is a dominant trait over green P's. Okay, so that's so smooth and yellow, but both are dominant traits. So the two traits are see the color and see the shape, right? If you cross the F1 plants, in the F2 generation, show all possible traits, including the recessive trait. Mendel called a cross involving two traits, so dihybrid crosses. All right, so two genes, two traits. You can also use a Punnett square to calculate the dihybrid cross. Here is an example, okay? The F1 plants have the double heterozygous, see here and here, okay? So in F2, what kind of phenotype and genotype do you expect to see? Many questions will ask you about the ratios. Punnett square is a detailed way to answer this type of questions. Just remember how to do the right combinations, okay? Here, this the big S can combine with either big Y or small y, okay, separately, because they are independent assorted, right? Similarly, small s can also pair with the big Y or small y, right? So based on this information, phenotypic and genotypic ratios can be calculated, showing in this Punnett square, okay? So this method is tedious, but very reliable, showing all these ratios here. The phenotypic and genotypic ratio of a dihybrid cross can be derived using a branched line method instead of a Punnett square. Okay, showing similar to this two. So this method is a little easier because you don't need to draw the square. Just kidding. Conceptually, it is the same as Punnett square. I will not go into details. If you don't understand the slides, please read your textbook. It has a very detailed description. Okay, here I would like to show you an easier way if the question only asks for the numbers, the ratio, okay? For instance, so here is the question. What is the number of all possible genotypes with dihybrid cross shown below? Always make sure that you are clear what the question is asking, right? So here it asks about genotype. I actually emphasize it by showing red here. But during the exam, there won't be any color, okay? If you go through the Punnett square or branch line analysis, I'm sure that you can get the answer correctly, but you won't have time left for other questions. An easy way is to think about the possible combinations in your mind. Here you see two traits, right? So then what about the possible genotype for each trait? Here shows possible phenotype for first trait has 3 possibilities, right? So big S, small s, or 2 small s's, right? Then how about second trait? Similar. You have 2 homozygous and 1 heterozygous. Based on this information, we learn combination in this mass, right? You learn position 1 has 3 possibilities. And then position 2 also has 3 possibilities. Then the total possible combinations are 3 times 3 equals 9. So the answer is 9. If you check the branch line analysis, indeed, 9 is the correct answer. Remember, this is for the genotypes, right? Okay, how about if I ask, what is the number of all possible phenotypes with a dihybrid cross shown below? The same method. You can pause and think. Okay, so instead of the genotype that each trait has 3 possibilities, now only 2 possible phenotypes for each trait. So therefore, the total number of all possible phenotype is 2 times 2 equals 4. Same as the branch line analysis shown below, all right? So the 4 different possibilities. Okay, so we are almost done with Mendel's law. This is a summary slide. We have discussed the 2 Mendel's laws, law of segregation and law of independent assortment. In some literatures, the law of dominance is also considered to be the 3rd Mendel's law. So this law suggests that some alleles are dominant while others are recessive. Yes, we learned that, right? So an organism with at least 1 dominant allele will display the effects of the dominant allele. Yes, with the examples of peas we learned, this is also true. However, in higher eukaryotic organisms, this is not always true. Let's take a look at several exceptions. We will discuss several scenarios in which alleles can interact in ways other than totally dominant or totally recessive. These include the incomplete dominance, meaning the heterozygous do not follow the phenotype of any parent, or codominance, meaning that both alleles are fully expressed in heterozygous, and multiple alleles, meaning 1 gene can have more than 2 allele options, okay? And in addition to these exceptions, scientists also found that expression of 1 gene may affect the expression of another gene. Epistasis is a good example. So I'll go through these ones one by one, okay? The color of a snapdragon flower is a good example of incomplete dominance. So red flower snapdragons and white flower snapdragons are both homozygous. When they are crossed, the F1 plant becomes heterozygous. Based on Mendel's law of dominance, the F1 generation should have a dominant phenotype. So either be red or white, depending which color is dominant, right? So interesting, the F1 you can see is a pink, meaning in heterozygous, the allele for red flower is incomplete dominant over the allele for white flowers. Therefore, the law of dominance does not really hold true in this example. Let's then talk about the multiple alleles. Our ABO blood types is a perfect example. I assume that you all know that ABO blood type contains type A, B, O, and AB, right? So, but you may not know that the blood types are controlled by 3 alleles. 2 of them are dominant and 1 is recessive. So dominant one is the IA and IB. The recessive is a small I, okay? But then, although there are 3 alleles available to choose from, we know that for each gene, every person can only have 2 alleles. One parent, one from the father paternal, and one from mother is a maternal. Therefore, the combination of the genotypes are more than what we learned from Mendele's piece, which only have 3 genotypes for each traits. So the ABO blood types can give totally 6 different genotypes and 4 different blood types, which is phenotype, right? So here I also need to clarify a concept, okay? So in this table, we know that the big IA, big A, this one, and a big I, 2BB, and the small I, those are homozygous. But how about this one is a big I with big A, a big I with big B. So both alleles are dominant, but they are different, right? So is this homozygous or heterozygous? So as we learned, heterozygous defines as different forms of genes in 2 alleles. So this is actually a heterozygous form, all right? In ABO blood type system, we can also see the example of co-dominance here. So cells expressing the IA allele, this is the big allele, they denote blood type A. Because the bigger IA allele produces these A antigens on the cell surface. And this A antigen is a glycoprotein, we learned that, right? In the second lecture, the cell membrane protein can work as a surface markers. Right? So similarly, these big IB denote blood type B by producing these B antigens, right? The recessive O type does not produce any surface antigen. Interesting in blood type AB, because both alleles are dominant. So both alleles are fully expressed, even it is heterozygous. So this phenomenon is called co-dominance. This had not been described in Mendele's theory. So genetics become more complicated in higher eukaryotic organisms. Lastly, let's just discuss about epistasis. Epistasis refers to the expression of one gene masks the expression of another gene. The most famous example of epistasis is called a Bombay phenotype. Also, it is known as the HH blood group or H antigen deficiency. It was initially discovered in Bombay, now known as Mumbai in India in 1952. This rare blood type is found in 1 out of 10,000 individuals in India and 1 in 1 million people in Europe. So although this is a rare blood type, for some reason, it frequently shows up in all kinds of genetics exams. So I figure I need to introduce this to you. Let's first take a look at the figure shown on the left, okay? You can see that there's a couple. Mom has a blood type A, and the suspected father has a blood type of AB. Why this dad is suspected of that? Because based on our knowledge, we learn from ABO blood types, the children should be any blood type, but not O, right? Therefore, whether this dad is the biological dad becomes suspicious. Then what happened is that we know the blood types are phenotypes. How about their genotypes? Interesting, the child's genotype is not this is small I as this usually all supposed to have. Instead, it's a big A or big B. Just for example, it can be AA as well. But why phenotypically it's not AB, the same as a suspected father? This is because our human being has another set of blood type system named the H blood type. Interestingly, the mom here is a heterozygous, big H, small h. The father is HH, heterozygous as well. And the child is a recessive homozygous, small hh. Okay. In this HH blood type system, the big H or the heterozygous HH, they make a protein called H antigen. Okay, so H antigen is found on virtually all red blood cells and is the building blocks for both A and B antigens. Without the expression of this H antigen in the surface of the red blood cells, and then no A or B antigens can be made because precursor, even this child that has a genotype of AB. So the inconsistency between the phenotype and genotype was not really explained in Mendel's law of dominance. So this is another exception. You can see when science gets advanced, we need to modify current hypothesis or theories based on the new discoveries. So always keep open-minded, okay? Okay, so let's practice several questions before I end this class. We will try several true or false questions and a few multiple choice questions. Lastly, I will go through 2 questions from our textbook. All right, so first of all, this first question, recessive traits are expressed only in the homozygous state. This is a true or false. Based on definition, a recessive trait is a trait that is expressed when an organism has 2 recessive traits. So this answer is true, right? Okay, see the second one. When analyzing a cross involving 2 traits, each trait is analyzed separately, and then the frequencies of each are combined to yield the observed phenotype ratios. True or false? This is exactly correct. And the meaning of independent assortment, okay? So the answer is true, all right? And the third one, incomplete dominance is the expression of a phenotype that is intermediate to those of the parents. Is this true or false? If you go back to check the snapdragon flower as example, you know this is the correct definition. Therefore, this answer is also true. Okay, this is a multiple choice questions. In a monohybrid cross with complete dominance, the F2 offspring should contain what? You can pause here. Okay, the correct answer is B. Okay, so this is a complete dominance, indicate it follows the Mendel's law, right? Monohybrid means only one pair of alleles are involved, such as a smooth P and wrinkled P. So we have always discussed this situation, so the answer is B. Okay, how about this one? The offspring F1 resulting from the cross between 2 red flowered plants, 3/4 red and 1/4 white. The most likely genotype both red flowers parental plants P1 is what? So the answer is D, heterozygous. The F1 in this question basically is the F2 generation in Mendel's experiment. So the red flowers can produce both red and white flowers in their offspring. indicating that red is a dominant trait and white is a recessive trait. So in addition, the P1 plants have to be both heterozygous in order to produce recessive offspring. So only D is the correct answer. Did you get it correct? Okay, so let's take a look at this one. So this is really easy. Mendel analyzed his pea plant data using the principle of what? So the correct answer is E. So this is very straightforward, and I guess everyone should get it correct, okay? Probability. Let's see this one, okay. Maybe you can read it and pause it here, read it to get the answer, and let's see what is the correct answer. Okay, so the correct answer is C. So the question needs a bit thinking here. I'll use the empty space as my scratch paper, okay? See here, I'll use this big H as the letter for hair texture gene, okay? So based on the information given in this question, here is a simple summary. So the big HH is curly. So the heterozygous is wavy, and the homozygous small h is straight, right? Then the question is asking about cross of HH and big H small h. Apparently, the phenotype of the offspring will be half big HH and half heterozygous H. And the question is asking about what? Phenotypic ratio. So then you know that you will have half curly and half wavy. So the correct answer is C. So a little bit of calculation will help you solve the question much better. All right, so let's practice a question in our test book. This is coming from page 66, question number 20. So our test book did not provide answer on this one. So take a look at it and may pause here, and then we'll do it together after you get the answer. All right, so the question is asking chance, basically the probability, this chance, right? Now I can use my scratch paper here to list this, okay? So if the question does not specify an exception, then defaultly we will consider Forest-Mendel's law, right? So we learned the law of independent assortment, indicating that we can calculate the probability of each gene separately, all right? Since you have learned the Mendel's P experiment so many times, I assume that you can solve this part easily. Remember, here the question is asking for the phenotype, all right? So to get this heterozygous AA, the probability is 50%, right? So to get this homozygous BB is 25%. And then this homozygous small c is again 25%. And this big heterozygous DD is half, 50%. And then homozygous E is 25%. So therefore, these 5 genotypes together, then multiply each other, is 1 out of 256. This is the final probability, and that is the answer for this question. All right, similarly, let's take a look at a question number 21 in the page 66. This is slightly different from question 20, all right? So this question is asking about phenotype, phenotypically. So you can calculate the probability of each phenotypes, this separately, right? So you can stop here to calculate. All right, so same strategy. So you calculate the probability of each phenotype separately, and you multiply this to get the overall probability. Okay, so showing here is the answer. So the answer multiple each gene together. This is 3 out of 8.
So once you learned how to solve these questions, you will be able to answer similar questions using the same way. For sure, we will test that in the exam, all right? So that's all for today. Thank you.